The next speaker is Viktoria Köppä from Stockholm University. Victoria is part of Linda Reicher's team. They have been very active in promoting and pushing forward the use of genetic diversity, NE, and especially indicators from Sweden. Some of you might know them. They're really, really active. And Victoria will tell us a bit more about the 9-country study they have conducted applying these indicators across the globe.
Thank you. As mentioned, my name is Viktoria Köppä, and I work at the Department of Zoology at Stockholm University together with Linda Reicher. And I came in a little bit in the middle of this process, but I had the great pleasure of being part of a large collaborative project to develop and test the genetic diversity indicators for the Convention on Biological Diversity. Just to recap, these are the indicators that we're talking about that have been adopted in the Kunming-Montreal framework. The NE500 indicator is the proportions of populations within species with an effective population size above 500. And the PM indicator, the proportion of populations maintained within species. So the NE500 indicator has been adopted as a headline indicator and the PM indicator as a complementary indicator. In this work, however, we also used a third indicator that was first suggested by the Coalition for Conservation Genetics, called genetic monitoring with DNA-based methods. And this is mainly a count of the number of species or populations that are being monitored with DNA methods. However, this was not adopted by the CBD then. This multinational pilot study included 9 countries across 6 continents. And the idea here was to capture, like Christina said, countries are diverse. So within these 9 countries, we have countries that have very different resources, access to different resources. We also have countries with quite different levels of biodiversity. We worked in teams of 2 to 10 people per country with 1 to 3 leads. And the goal was to assess 50 to 100 species per country. And we worked quite in strong connection with personnel in different biodiversity agencies, and altogether there were quite a lot of people that contributed to this process in different aspects, either by helping out with data, helping out with expert consultations on assessments and so on. And this was quite collaborative participatory process. As mentioned, this work started long before the CBD was adopted, and the idea was to test this out and also during the process refine methods and so on. So it's quite a long iterative process. Together, we evaluated a total of 909 species across several taxonomic groups, which amounted to roughly 5,000 populations assessed. On average, each assessment took 2 to 3 hours. And I'm going to jump straight into the results first here, a bit unorthodox perhaps, and then later on we can talk a little bit about the underlying data and the methods. But we found that for the majority of species examined, they have an indicator value of 0, which means that all their populations are too small to retain genetic diversity. And that we can also see that the pattern is the same for both animals and plants. For the PM indicator, on the other hand, we found that the majority of species examined maintain all of their populations. One possible reason for this is the lack of available information on species occurrence and populations further back in time, which makes it difficult to identify lost populations. In addition, we found that the pattern of species indicator values across the global red list categories are quite similar. And even among species then that are listed as least concerned or near threatened in the global red list, have many populations that are experiencing genetic losses because their populations are too small. When it comes to then the DNA-based genetic monitoring indicator, we see these are the numbers for the number of species in each of these countries that have monitoring. Sweden is in the lead, yay. And one of the things that is important to highlight here then is that even if we find that the proxy data is very useful and it's possible to do these assessments with proxy data and the first two indicators, they don't necessarily require DNA data. We think it's also important to highlight that we shouldn't forget that we also need to do DNA-based monitoring because they also capture other processes. So let's not stop with the DNA-based monitoring just because we have good proxy data that we can use for the indicators. So if we dive a little bit back into the underlying data, then I think Christina already mentioned a lot of these sources. We used already available data to use these assessments. For example, national databases with red list data or official reports, such as species recovery plans, management reports, distribution data from different sources, from inventories or habitat quality or citizen science. We consulted scientific papers, discussed with experts, and in a lot of cases, especially in countries where they don't necessarily have the national resources that we do in a lot of Nordic countries, there was consultations with local knowledge holders. To aid in this data collection, we used an open software tool called Kobo Toolbox, where you can create and administer projects and create questionnaire forms, online interactive questionnaire forms, where you can have a controlled vocabulary. That means that you can control for how people answer the questions. And the reason for this is because when you're going to have 50 different people who are going to collect data, if you ask them to do this in an Excel sheet, you're going to get a lot of potential errors, and it's going to be a lot of work trying to put all that data together for analysis. So what type of data are we talking about then? What type of data do we actually need to calculate indicators? Remember again that the indicator value is based on a proportion. So the proportions of populations maintained or the proportions of populations with effective size about 500, where then an indicator value of 0 is the worst case scenario for all the populations. And an indicator value of 1 is the best scenario. We have this example here on how to calculate this. So for the NA500 indicator, so in this case, we have 3 populations and one of them are above 500, and then we have an indicator value of 0.33, the proportion. And for the PM indicator, then we have 1 population that is lost. So in total, before we had 4 populations, and now we only have 3 populations maintained, 0.75. So the information really that you need to calculate these indicators are the number of populations and census size in each population and the number of lost populations. Seems simple enough, right? Which brings us to sort of the hot potato or the golden question of the day. What is a population? This is one of the most common questions that we get. And it's also one of the major challenges that we faced during this project. And one way to that we dealt with this, because since we're working with already available data, was the idea we kind of have to work with what we have, right? So we decided to be quite flexible in how we collected this data and how we allowed people to define populations in order to be able to do this. So depending on what information you have available for your focal species, there are several ways that you can go about this. And hopefully there might be some type of genetic studies where there's been structural analysis or something similar where you can define distinct populations based on genetic data, and then you can use that. In other cases, you might be able to use phenology or morphology to divide them into units, such as ecotypes, as an example of that. Or there might be ecological or biogeographic proxies, such as habitat zones that you can use, or management units. And often a combination of different methods needs to be used depending on what information is available. So in our questionnaire form here, we allowed for the assessors to tick boxes then of what method have they used to identify these populations or combinations of methods. And further down also an explanation or possibly a map of how this was done. So here is an example from our study. This might look familiar to some people in the room, of where we have used genetic differences or clades or clusters to identify populations. Brown bear in Sweden then, where 3 clusters have been identified, the northern, middle, and southern cluster. It's important to highlight here, though, that these clusters are transboundary. They're shared with Norway, which is an issue that one might want to consider also in the national reporting. In this case, it's not a huge problem since the majority of the populations are found in Sweden. So, and also this species is they're well below the threshold, anyways. But in cases where you have these type of transboundary populations, and you try to decide how many of these, what is the effective size of your country's responsibility, this becomes a challenge. Here's another example of using ecological or biogeographical proxies instead. So these are eco regions that have been used in the United States. Here's another example where we have used, so this is from Belgium. This is work from Joachim, who used a method that we decided to call dispersal buffer, where you use occurrence data and known dispersible distance to try and identify clusters this way. So a lot of this methods, a lot of work in this project went into developing these types of methods and answering these questions on how to go about this, and designing guidelines, both for the web form, but for in general, how to assess the indicators. And these guidelines are available online, along with also a ready-to-use template for this web form that you can use. We also have a GitHub project dedicated to the genetic diversity indicators, with ready-to-use scripts available for the estimation of the genetic diversity indicators, and used based on the variables that are collected through this COBO form, because it's important to highlight here that the COBO form only collects the data. It doesn't calculate anything, so it only and then you can extract the Excel sheets, and then we have a script that does the actual calculations. So one of these questions that came up early in this project was, do we have enough data available to do this? Do species have data available? And so this is an on number of populations done. So we found that most species have data available on the how many populations we can define. But this is also biased towards that you could be very flexible in how you identify populations, right? So the data is not necessarily presented in a very straightforward way. But most assessors felt that they were able to identify the number of current populations. If we, however, look at the available data on extinct populations, this was a much bigger challenge, as you can see. So that the orange bars here are the number of the unknown extinct populations. So on cases where for individual species, the assessor answered, I cannot determine the number of extinct populations. When it comes to the census data, so we preferably want data on population level census size, right? And this is usually not necessarily the case when you look at national reports or so on, like this, the red list reports on the national level. So the green bars here are those species that had information on population level, and the blue is when it was only on national level, on the whole species level then. And the gray, I don't know if it's possible to see all the gray, but that's insufficient data. So census sizes is not always available. We also allowed for census size to be collected in different ways. So one thing to highlight here then is that it's a combination of both proxy and genetic data sometimes, because when there are any estimates available from genetic data, we use that. And out of all the species done, the ones in green then had any estimates available. Then we allowed for census data to be collected in two forms, either as point estimates or as range estimates, where perhaps an expert might be able to say in this area there are more than 5,000 individuals or much less or so on. And as you can see from the color scheme here, that was the most common form of the census data that we had on our data set. Overall, though, we found that over 80% of the species assessed had data for at least one indicator. It's also possible that the census size is not collected in the same way for all the populations of one species. There might be different types of census data available for the same species. So it's collected per population then. So it's also possible if you have any data for one of the populations, you can use that and then use census size for the other populations. What about uncertainty, you say? We also allowed for this COBO-form system, allows so each COBO-form has a unique ID, which means that you can collect COBO-forms, several COBO-forms for the same species. This allowed us to do multiple assessments for species where there was a large degree of uncertainty. For example, you might have one expert who says, well, this should be considered one population, and another expert thinks this should be considered two populations. Then you can do individual assessment for both those alternatives, calculate the indicator value, and then take an average. And overall, we found that when doing this for some individual species, there is a large difference in the indicator value, but in most cases, this only resulted in a very small change, the difference. We also examined the effect of using other NE to NC ratios, because this is also a common question that you have gotten. Is it really feasible to use 0.1? And we can see that in some cases it does have a large effect on the individual value for one species, but it doesn't have a large effect on the overall pattern and the overall indicator values. Those of you who were at the workshop yesterday will recognize this picture. It's made from Belgium, where they have examined a frog and compared the estimation that comes from proxy data and the estimation that comes based on genetic data. And we can see that there's a large overlap in this. So for when the DNA was used, they identified that 4 out of 24 populations had an NE500 size above NE500, which gives an indicator value then of 0.125. And for the proxy data, this was 3 out of 24 populations then. So an indicator value of 0.166. So we can see that it has quite a good overlap. Finally, I just want to highlight a little bit of the work that has been done in Sweden. So this is my part of this large study. In Sweden, we have evaluated 120 species with the arrows have moved, but 2 full groups that we have examined, amphibians and reptiles. It should move down. And I discussed this yesterday. The reason that we were able to do this is also that we have 13 amphibians and 7 reptiles in Sweden. So not an enormous task to do full groups for those species groups. But we can see that the overall pattern is the same for the Swedish results. There has also been a pilot study made in Sweden at the beginning of this project that used only red list data by Henrik Turfjell, and together with Linda and more people. And they examined if it was enough data available in the red list to do these assessments, and found that 33% of species in the Swedish red list had data available for the first indicator, and 20% have had data available for the second indicator, and that 0.3% of the species in the Swedish red list are monitored genetically. They also made rough indicator assessments for 3 groups, mammals, amphibians, and reptiles. So one of the things that we did in Sweden following this 9 countries assessment is that we compared the first and the second assessment. And the difference between these 2 assessments is that the first one is based solely on red list data, just sort of quick and dirty. And the second assessment then has used several sources and also looked at occurrence maps and so on, and gone over a little bit more in detail. And what we found from this is that in total there were 37 species that were included in both studies. But we only compared the PM indicator for a subset of these because the methods for evaluating these was too different. But for the NE500 indicator, there was a difference in the assessment value in 23 out of 37 species. But the average difference in the indicator value was quite small. For the PM indicator, there was a difference in 14 out of 23 compared. And again, the difference was quite small. So what we can tell from this, I mean, you can see that this is significant for mammals, which is something interesting to consider. But one of the things that I want to highlight with this is from the discussion that we had yesterday of how much work do we need to put into making these assessments for the national reporting if we want to look at large trends. Again, this goes into the discussion about them being imperfect or about uncertainty, that how much work do we need to put in to get a good estimation of the large trends? Yes, for individual species, it matters a lot, but for the overall patterns, it doesn't. Another point that I wanted to bring up from our work in Sweden is that a large portion of our data does come from the Swedish Red List, even in the second assessment. And the first pilot study shows that there is a lot of data available in the Red List database to assess the genetic diversity indicators. And several criteria within the Red List use parameters that are related to the information that you need in order to assess the genetic diversity indicator, such as we have here, the number of subpopulations, the number of mature individuals, and the size of the largest subpopulation. So one of the discussions we have, this table, by the way, comes from the IUCN guidelines. And so one of the discussions that we had in Sweden was if it would be possible to do the assessment process in parallel with the Red List process, since they're already looking at the same type of parameters. So therefore, we carried out interviews with Red List managers to hear their opinion on this possibility. And we found that 8 out of 12 interviewed managers were very positive to this idea, whereas 4 out of the 12 interviewed managers were a lot more skeptical. And we don't really have time to go over all of the comments here, but the challenges that they raised, that the managers that were more skeptical, are often related to their taxonomic group that they're working with, that there are challenges with available information, or you have high lot of fluctuating population numbers and so on, which makes this a lot more challenging. The take-home message then from today or from this study is that calculating the genetic diversity indicators, it is possible and it is feasible. We managed to evaluate over 900 species and 5,000 populations, so there is data available. Over 80% of the species had data for at least one indicator. Overall, our results show that most populations are too small to maintain genetic diversity. And most species have the majority of their population maintained for now, but we need to prevent further losses, of course. Here, I would like to give my thanks to all people who have been involved in this. When Elisa gives this talk, she usually refers to her small army of collaborators, which is very true. There's been a lot of people involved in this work. And as Christina mentioned, a lot of organizations. With that, I would like to say thank you for this opportunity to come here and present this work, and thank you for listening.
Thank you very much, Victoria. Running a bit late, but I think we should take a few questions if they are. Also to Christina's talk. Okay.
Thanks for a nice summary. And it's a bit dispiriting if the conclusion is that most populations are way below the limit. But I guess to come back to Robin's talk earlier, how are you incorporating some degree of gene flow into this? Because as we heard earlier, then actually just a small amount of dispersal among these populations could quite dramatically change the conclusion, right?
That's okay. I mean, obviously, that is an issue that we very much discuss, and it goes into this questions of how do we identify the unit? In our case, this unit that we want to look at is the populations, and we try to use the definition that IUCN has for subpopulations as well. So ideally, they're not completely isolated. They allow for some degree of gene flow, but we try to identify what you would consider the genetic, the true population in those terms. And in those cases where you have a lot of functioning metapopulation, that you would look at the metapopulation level in that case. However, of course, in this work, when we use available data and we use these types of flexible methods to identify our units, this is going to be very much imperfect in that respect. Absolutely.
So, of course, I was thinking of gene flow as well as Henrik. So it seems that most of your data come from endangered population. Did you manage to actually get data for just normal population and compare the index that you find between non-endangered and endangered? Because for me, and it's what Jane said, population of 500 individuals, any of 500 in a metapopulation seems to be what most of animals or plants are doing. So I would say, how do we know if these index are good enough or are sensitive enough if we compare them to not endangered population species?
And by populations, then, do you mean the populations within a country, the IUCN subpopulations, or do you mean population as a whole?
Type of species, because if you talk about a fox will be totally different from a frog, which will be totally different from a fish.
So I can answer starting with the first questions on how this bias in our data set towards endangered species, which I talked about with Robin this during the short break too, that there is a bias in our data set in the sense that the results that we have are calculated based on the species that had available data. Obviously, and species that tend to have available data are species that are of great concern in some way. There could be species that have potentially large populations and so on because you collect data on them because they're commercially important, and those species we have data on as well. Species that are well managed, for example, a lot of maybe large predators and so on, also have data available. So in that sense, there will inevitably be a bias towards species that are endangered in some way or another, because those are the ones that we tend to monitor. We tried as much as possible to include a broad range of species across, and have species of least concern and so on included in the data sets. We also try to include different taxonomic groups, different realms, different types of life histories, and so on. So we try to account for those types of differences. But I think it's inevitable to run into those types of problems. But when it comes to comparing endangered and non-endangered populations, the indicator value to begin with, or the data, is collected on a population level, right? So if you have a species and you're going to take data on all of its populations, and you do that within the nation's boundaries, you're going to have all the different values from the different populations, and you can compare that. It might be interesting to look at the same species for different countries, if same countries would collect on the same species and see what are their indicator values. I think that's quite interesting. We have a few examples, at least in our data set in Sweden, where I know that we have species which have very different, that have some very large populations and some very small populations. It's not included in our data set, but a classic example from Sweden is a bird that's very common up, it's capercaillie. So it's very common up north in Sweden and has more or less a continuous population in the north of Sweden. And in the south of Sweden, it's found in isolated patches. So that's a great example of one of those species where you can find very different situations for different populations. Does that answer your question, you feel? Yeah.